Good day, everyone. Today we will discuss one of the most important processes in human physiology, muscle contraction, specifically through the sliding filament theory and the cross-bridge cycle.
Every time we walk, blink, smile, or even breathe, our muscles are contracting. But what actually happens inside our muscle cells that allows movement to occur? In this microlecture, we will explain what the sliding filament theory is, how the cross-bridge cycle works, and how these processes produce muscle movement.
By the end of this discussion, you should clearly understand how muscles convert chemical energy into mechanical force.
Each muscle contains muscle fibers, and inside each muscle fibers are multiple myofibrils. These myofibrils are long cylindrical structures composed of repeating sections called sarcomeres. Myofibrils consist of even finer fibers called myofilaments, which are of two types: thick filaments and thin filaments. Thick filaments are made of a protein called myosin, while thin filaments consist mainly of actin. The specific arrangement of actin and myosin gives skeletal muscle its striated appearance.
Let us now discuss the structure of actin and myosin in detail. Each thick filament consists of hundreds of myosin molecules stuck together with the myosin heads facing outward. A myosin molecule is shaped like a golf club with a globular head and a long shaft-like tail. The thin filaments, on the other hand, consist of two intertwined chains of the contractile protein actin, forming a structure that resembles a string of beads.
Associated with actin are two regulatory proteins, tropomyosin and troponin. These proteins play opposite roles in muscle contraction. Troponin promotes binding between actin and myosin, facilitating contraction, while tropomyosin blocks the myosin binding sites on actin, preventing contraction. You can remember the function of tropomyosin as stop myosin since it prevents actin-myosin interaction. In the sarcomere, the Z line is attached to actin while the M line is associated with myosin. You can remember this as M for myosin and M for M line. During contraction, the thin filaments are pulled towards the center by the thick filaments, increasing the overlap between actin and myosin. As a result, the Z lines move closer together, shortening the sarcomere. This is the sliding filament theory.
To make the structure of the sarcomere easier to remember, consider this simple guide. The sarcomere is the basic contractile unit of the muscle fiber, and to draw a sarcomere, first, draw two Z lines which represent the borders of the sarcomere. Next, draw an M line in the middle, then add a dark A band representing the thick myosin filaments. Finally, the remaining lighter regions are the I bands representing the thin actin filaments. Now let us understand the process of muscle contraction step by step through the cross-bridge cycle.
Step 1. Motor neuron action potential and acetylcholine release. An action potential reaches the presynaptic terminal, opening voltage-gated calcium channels and inducing the release of acetylcholine. Step 2. Acetylcholine binding and motor end plate depolarization. Acetylcholine binds to receptors on the postsynaptic membrane, leading to depolarization of the muscle cell membrane at the motor end plate. Step 3. Propagation of action potential. The depolarization spreads across the muscle fiber and deep into the cell through the T tubules. Step 4. Calcium release. Membrane depolarization causes conformational changes in the voltage-sensitive dihydropyridine receptors, which are mechanically coupled to ryanodine receptors on the sarcoplasmic reticulum, leading to the release of calcium in the cytoplasm. Step 5. Exposure of myosin binding sites. Calcium binds to troponin C, causing tropomyosin to shift and expose the myosin binding sites on actin. Step 6. Cross-bridge formation. The myosin head binds strongly to actin, forming a cross-bridge. ATP is hydrolyzed into ADP and inorganic phosphate. Step 7. Power stroke. The release of inorganic phosphate initiates the power stroke, during which the myosin head pulls the thin filaments towards the center of the sarcomere, producing force and shortening the muscle. ADP is released at the end of this step. Step 8. Cross-bridge detachment. Binding of a new ATP molecule causes the detachment of myosin head from actin. Step 9. Cocking. ATP is hydrolyzed into ADP and inorganic phosphate, returning the myosin head to its high-energy cocked position, allowing it to bind to a new site on actin if calcium remains present.
Calcium is then actively reabsorbed into the sarcoplasmic reticulum by calcium ATPase pumps, leading to muscle relaxation. Now, to visualize clearly using moving animation, calcium binds to troponin, shifting tropomyosin and exposing actin binding sites. Myosin heads attach and perform power strokes using ATP, then slides actin filaments inward, shortening the sarcomere. And when the calcium is pumped back, the muscle relaxes. Now that we understand how muscle contraction works at the cellular level, let us see how this process is applied in real life.
Every voluntary movement depends on the coordinated contraction of skeletal muscles. In sports and exercise, repeated cross-bridge cycling allows athletes to generate force, speed, and endurance. Muscle contraction is also essential for vital involuntary functions. The heartbeat relies on rhythmic contraction of cardiac muscles, while breathing depends on the contraction and relaxation of the diaphragm and intercostal muscles.
To summarize, muscle contraction occurs through the coordinated interaction of actin and myosin, guided by the sliding filament theory and driven by the cross-bridge cycle. The release of calcium in a continuous supply of ATP allows myosin heads to repeatedly bind, pull, and detach from actin, producing force and movement. This microscopic process enables every action we perform, from simple blinking to powerful athletic performance and essential life-sustaining functions such as breathing and heartbeat. And by understanding how muscles contract at cellular level, we gain deeper insight into human movement, exercise performance, and clinical applications in health and disease. Thank you for watching, and I hope this microlecture helped you clearly understand the science behind muscle contraction. Bye!